Rochelle Crosbie Watson, and today we're going to learn about how the dystrophin-associated protein complex was originally discovered. So I'm a protein biochemist, and this topic is particularly exciting to me, and I hope you enjoy it too. So as you remember, dystrophin is associated with a complex of proteins called the Dystrophin-Glycoprotein complex. Now, this complex didn't just appear out of thin air. Scientists worked very diligently to discover each of the components of the dystrophin-glycoprotein complex in order to come up with the model that you see here. So what we'll discover today are the steps of the purification that led to the discovery of this complex of proteins. In addition, we'll find out how the discovery of this complex led to further other scientific discoveries and the identification of other forms of muscular dystrophy. So let me show you the purified complex first. This is going to be our end game, if you will. Shown on the left is an immunoblot. It's stained with antibodies to all the components of the dystrophin-glycoprotein complex, except for sarcospan. The immunoblot on the right is stained specifically with antibodies to sarcospan. In lane 1, you can see that the sample is crude surface membranes isolated from rabbit skeletal muscle. And then in the second lane, purified dystrophin-glycoprotein complex is analyzed. And I hope it's easy to appreciate the dystrophin, which migrates at about 427 kilodaltons. Remember that it's a really large protein. The dystrophin is enriched during the purification. Similarly, all the proteins that are associated with dystrophin, like the dystroglycans shown in pink in the schematic diagram, and the sarcoglycans shown in yellow in the schematic diagram, are also enriched during the purification. So alpha-dystroglycan migrates at about 156 kilodaltons. It appears as sort of a broad smear because it's heavily decorated with carbohydrate residues. The other proteins, which we'll learn later on, are also modified with carbohydrates, and they migrate on the gel anywhere from 25 kilodaltons to about 50 kilodaltons, which you can see in the immunoblot, in which all the antibodies are stained with the pan-DGC antibody. So this is our end game. This represents the starting material, the crude surface membranes, and the ending material, the purified dystrophin-glycoprotein complex. But there were many steps that happened in between there, and we're going to learn exactly what those steps were today. So before we get started, let me introduce you to some of the basics regarding protein purification. So there are many, many different ways to purify protein. And biochemists traditionally use sucrose or cellulose beads that are coupled to other molecules that can be used to affinity purify proteins from a large sample or a large homogenate that contains many, many, many different proteins from a given sample. Like for instance, today we're going to be looking at whole skeletal muscle or even crude surface membranes, which represents all the surface membranes from the skeletal muscle. So in this particular sample, we are going to be looking at a lectin affinity column. And I'm showing you with the little triangle cutouts where the affinity sites are located, or where the lectin is coupled onto the sucrose beads. So first, a whole starting material sample is applied to the column. Only the proteins that bind specifically to the lectin will be retained onto the column. The other proteins flow through in a void sample. And then a competitive molecule, like another lectin, is added to the column, which competes off this specific protein, and that protein sample can be collected as an eluate in many different fractions. In addition, those proteins, the starting material, the flow-through or the void, which represents the unbound materials, the bound materials, which are found in the eluate or the purified sample, can be analyzed. Usually they're analyzed by SDS polyacrylamide gel electrophoresis, which is a process in which proteins are denatured and reduced using a reducing agent like beta-mercaptoethanol. They're applied to a polyacrylamide gel, which separates proteins using electrophoresis based on molecular mass. And then proteins are analyzed by Kamasi blue, which reveals all of the proteins because the stain binds to the polypeptide backbone. The gels can also be analyzed using specific antibodies that recognize an epitope or many epitopes on a given polypeptide protein. So for instance, the gel material can be applied by electrophoresis to a nitrocellulose or a PDF membrane, and the proteins are transferred using electrophoresis to this blotting membrane. The membrane then can be probed with specific polyclonal or monoclonal antibodies to detect that protein of interest. And so we're going to be looking at many Kamasi-stained protein gels and many immunoblots, also called Western blots. And you may have heard about this technique in some of your other classes. So we're going to discover today the paper that was published in 1989 in Nature by Kevin Campbell and Steve Kale. And in this paper, they document the steps needed to purify dystrophin and its associated complex. This is really a wonderful, groundbreaking paper, and I'm really excited to present it to you today. What's kind of neat is in the abstract, they write, our results indicate that the function of dystrophin could be to link this glycoprotein to the underlying cytoskeleton and thus help preserve membrane stability. And they were right on the money. The function of dystrophin and the associated glycoprotein complex is to protect the sarcolemma from contraction-induced injury. So I really love that they nailed the function of this complex in their first groundbreaking paper. So this project was brokered by the Muscular Dystrophy Association. And at the time, Lou Kunkel at Harvard University had just discovered that mutations in the dystrophin gene cause Duchenne muscular dystrophy. That in itself was a groundbreaking discovery. At the same time, Dr. Kevin Campbell at the University of Iowa was working on voltage-gated calcium channels. And in fact, his lab had wonderful expertise in purifying these calcium channels from skeletal muscle and also from other tissues. That's no small feat. Calcium channels are comprised of large subunits that are connected together within the membrane bilayer. They're glycoproteins, and they're quite difficult to extract from the membrane. One has to use very gentle detergents and very gentle conditions. And his lab had developed expertise in this area. And so the Muscular Dystrophy Association was very keen to understand the function of dystrophin at the protein level and interested in encouraging scientists to investigate that. And so they brokered this collaboration in which Lou Kunkel shared some of his antibodies with Kevin Campbell so that Kevin Campbell could use those antibodies as a molecular tool and look in his purified samples and ask the question, do we ever see dystrophin as part of our purification? So we are going to look at the steps of the purification and discover how Kevin discovered dystrophin and its associated glycoprotein complex. So, step one of the purification process. Now, this purification was in large part designed to purify calcium channels from skeletal muscle membranes. And you will see that throughout the steps of the purification, where we will be analyzing exactly what happens to specific skeletal muscle proteins, and in particular, what happens to the calcium channel during purification. So skeletal muscle membranes are prepared usually from rabbit skeletal muscle, just because one can obtain a lot of tissue from rabbit, although this procedure can be done really with any skeletal muscle. The skeletal muscle membranes are gently solubilized with a detergent called digitonin. Digitonin has a chemical structure that's similar to cholesterol. There is also some salt that's added to the solubilization. That solubilized sample is then applied to a lectin column. In this case, Dr. Campbell is using a succinylated SWGA column. That column is then washed numerous times. The void or the flow-through is collected, and the eluates are collected. And the eluates are collected by collecting fractions 1 through 12. So the first fraction that comes off the column is called fraction number 1, and the last fraction is called fraction number 12. And in this case, because the lectin is SWGA, the molecule that's used, or the lectin that's used for elution is N-acetylglucosamine. So what you'll see in the experiments provided here is a Kamasi stained gel, and this gel is revealing to us all the proteins that are present in the sample. Lane 1 is showing us the starting material. So the starting material consists of a lot of proteins, and we've just are showing you a few of them here, like nebulin and ranitidine receptor, myosin, as well as the ATPase, okay, calsequestrin, lots of proteins. And the flow-through represents the proteins that are not binding to the SWGA lectin column. Okay, so everything that's not sticking to the column just comes right out in the flow-through, also called the void. You're also seeing an immunoblot of dystrophin. And what I hope you can see is that in the starting material, dystrophin is very faintly present. And in fact, if you look at the whole homogenate of skeletal muscle, dystrophin is not really a robustly expressed protein. So when you look at the total proteins, unlike myosin and actin, which represent a large majority of the proteins that are found in skeletal muscle, dystrophin is a lowly abundant protein. And that's suggested by the fact that it's staining very weakly in the starting material, or lane 1. Now, did dystrophin stick or bind to the SWGA column? The answer is probably so, because we're not seeing it in the void or the flow-through, so probably it's stuck to the column. Here is an example of what I just showed you for an SWGA enrichment. The skeletal muscle membranes are applied to an SWGA separose column. That column is washed. The void is analyzed by Kamasi stain gel and by immunoblot. The column is eluted with N-acetylglucosamine, and fractions are collected. This next slide is showing us an analysis of those fractions. So you'll see that the gels are analyzed by Kamasi blue stain, which is representing all the proteins that are part of that SWGA eluate. And then there's the dystrophin immunoblot, which is looking specifically at the dystrophin protein. Again, remember, the dystrophin protein is huge. It's 427 kilodaltons, so it migrates really high on the SDS polyacrylamide gel. So what I hope you can see is that dystrophin is occurring in the earlier fractions. It's eluting from the SWGA column in those earlier fractions. And similarly, the channels, the calcium channels, are also eluting in the same fraction. So what this is suggesting to us is that dystrophin itself might be a glycoprotein and binding directly to the lectin, or dystrophin could be associated with other glycoproteins that are being retained on the SWGA column. So this first purification got rid of a lot of proteins. It got rid of nebulin and myosin and other cytoskeletal proteins that are part of the contractile apparatus, but it did not separate out the calcium channels from dystrophin because they are found in very, very similar, they're found in identical fractions, as you can see here. So the next step of the purification is a DEAE anion exchange column. And in this step of the purification, the fractions that contained dystrophin, those SWGA eluate fractions, are collected and they're loaded and applied to a DEAE anion exchange column. Again, the proteins that are not bound to the column are flowed through and collected and analyzed. The column is then washed, and then the DEAE anion exchange column is eluted with sodium chloride. Remember that this is a charged cellulose column, and so eluting with sodium chloride is the appropriate way to elute specifically binding proteins from that column. So the column is eluted with 50 millimolar sodium chloride, 100 millimolar sodium chloride, and so forth and so on, all the way up to 250 millimolar sodium chloride. And so again, those fractions are collected. And whenever you're doing any kind of protein biochemistry experiment, you really want to analyze all steps of the purification. You want to analyze the material that's applied to the column. You want to analyze the flow through of the void, which is why I keep coming back to this flow through and void. You want to analyze potentially even the wash fractions, as well as all the specifically eluted fractions, because you want to trace where is my protein during the purification. What happens? What's the fate of my protein during the purification? So step 2 of the purification was this anion exchange column, and the results are shown here. So I'm going to take you through it. Again, just like all the other analysis that we've seen before, we're looking at SDS polyacrylamide gels that are stained with Kamasi to reveal all the proteins, and we're also looking at an immunoblot stained specifically with the dystrophin antibody, so we can really find which of those proteins is dystrophin. So let's look at lane 1, and you can see that lane 1 is our starting material. Basically, lane 1 is a collection of those SWGA eluate fractions that contain the calcium channels, and they also contain dystrophin, okay? And you can see dystrophin on the Kamasi stained gel. It's very, very faintly present at around 400 kilodaltons. And then the alpha 1, the alpha 2, the beta, and the gamma are subunits of the calcium channel that are present in that starting material that's applied to the DEAE anion exchange column. So the first step of the elution is a 50 millimolar salt wash, and the laboratory made the decision to collect 2 fractions of that salt wash, a fraction 1 and a fraction 2, and they analyzed each of those fractions. And I hope you can see, it's easy to see looking at the Kamasi stained gel, that the calcium channels come out in that low salt elution. But dystrophin, if you look at the immunoblot, you don't see any dystrophin. Aha. This is the first suggestion that we've separated out biochemically the calcium channel from the dystrophin protein and potentially any other proteins that could be associated with dystrophin. In the second elution of the DEAE anion exchange column, which is 100 millimolar sodium chloride, again, they elected to take fraction 1 and fraction 2 of 100 millimolar sodium chloride elution, and you can see that even the calcium channels are bleeding off a little bit more. We're releasing more of those calcium channels by increasing the salt concentration just a little bit. If you look at the corresponding immunoblot, there is no dystrophin, again suggesting that we're separating out by using different concentrations of sodium chloride. We're separating out the calcium channels from the dystrophin protein. Aha. The piece de resistance, the 250 millimolar salt elution of the DEAE anion exchange column reveals very, very low levels of the calcium channel. Some present, but very, very low levels of calcium channel. But all of the dystrophin is eluted in the first fraction of the 250 millimolar salt elution from this column, which is the first evidence that we've purified dystrophin and potentially any other proteins that dystrophin is interacting with. So Dr. Campbell and Dr. Kale wanted to further purify dystrophin and any other proteins that could be interacting with it. And to do that, they used ultracentrifugation. And ultracentrifugation is a method that really separates protein complexes so that proteins that are not bound in the same complex will be found in different fractions. Let me show you what I mean. So what they did is they took the 250 millimolar salt eluate from the prior purification step, and they applied it to the top of a sucrose gradient. Now this is basically a large test tube that's loaded with a gradient of sucrose from 5% to 20%. And then that gradient is spun by ultracentrifugation at really, really high speeds. Proteins are separated, as you can see schematically here. They're separated based on molecular mass and frictional coefficient. Frictional coefficient is basically you can think of it as the shape of a molecule or the shape of a complex of proteins. And proteins that are bound together tightly in a complex will be retained in the same fractions during the centrifugation. So this is a very rigorous way to look at protein complexes and protein-protein interactions. And then finally, after the centrifugation, so this will be the last step where we have proteins that are separated into distinct layers within this test tube gradient. Fractions are collected from the top of the gradient all the way down to the bottom of the gradient, so that the first fraction that's collected is called fraction number 1, 2, 3, 4, and so forth and so on, down to the bottom of the test tube, which represents the 20% sucrose, and that's fraction number 12. So fraction number 1 represents the low percentage sucrose, and fraction number 12 represents the higher percentage sucrose. Okay, so when we analyze those protein complexes, those protein samples, proteins that are found in a complex will migrate in the same fraction. So this SDS polyacrylamide gel is stained with Kamasi, and in this particular gel, we're looking at an analysis of all the fractions that are collected from the sucrose gradient centrifugation, such that the lower fractions represent the lower percent sucrose gradient, and the higher fractions represent the bottom of the centrifuge tube, which is the higher percentage of sucrose. And what I hope you can see is that the fractions that are outlined with the red box represent dystrophin, and what we can conclude is that those other proteins that are migrating in the lower part of the gel, and they migrate between 25 and 50 kilodaltons, those proteins are associated in a complex with dystrophin. And they're pulled away from a very small amount of sodium channel, voltage-sensitive sodium channels, that happen to slightly co-purify or kind of contaminate the purification of dystrophin. So this is a very sensitive, very rigorous method to really pull apart protein complexes. And what Dr. Campbell concluded from this is that the proteins outlined in red constitute a complex. That complex represents proteins that are interacting with dystrophin. Now, we don't know the identity of those other proteins, okay? We know their molecular mass. And I just want to point out that it's possible that there could be more than one type of protein or type of polypeptide at a given molecular mass, okay? And those discoveries, I'm kind of giving you the little foreshadowing, those discoveries were made in the decade that followed this groundbreaking experiment. So one of the experiments that Dr. Campbell and his postdoc at the time, Dr. Avasti, asked was, well, we know that dystrophin, which causes Duchenne muscular dystrophy, we know that it was retained on the SWGA column, which is a lectin affinity column, during the very first step of the purification. It was not found in the void or the flow-through. And SWGA is a lectin that binds to a pattern of carbohydrates that decorate proteins. Okay, fine. Does that mean that dystrophin itself is a glycoprotein, or does dystrophin associate with other proteins that themselves are glycoproteins and are retained on the SWGA column? So to get at that, they performed an experiment in which they took nitrocellulose membranes and they incubated them with WGA peroxidase. So basically a lectin that binds to glycoproteins, and they developed that in order to visualize the proteins that are glycoproteins. And I've lined it up side by side with the Kamasi stained gel of the purified dystrophin-glycoprotein complex. So outlined in blue, you'll see the Kamasi of the purified dystrophin, and it is not being recognized by the WGA because there's no binding of WGA to dystrophin. That's the first evidence that dystrophin itself is not a glycoprotein. Let's contrast that to the protein that I've outlined in red, which migrates as a kind of a large smear at 156 kilodaltons. It doesn't pick up Kamasi stain very well, but it's definitely present. It stains very, very well with silver staining, which is another very sensitive method for staining proteins. And you can see that the WGA binds very, very strongly to this 156 kilodalton protein, which suggests that it is definitely a glycoprotein of some type. Do we know the identity of that protein? No, absolutely not. But it could explain why dystrophin is retained on the SWGA column during the first step of the purification. I also just want to bring your attention to the lower part of the gel. You can see that the WGA is picking up a few of those other glycoproteins as well, suggesting that dystrophin is associated with one or more glycoprotein. And that turns out to be right on the money. Okay, one of the brilliant things that Dr. Campbell did once he purified dystrophin and its associated glycoproteins is he created molecular tools. In particular, he created a whole plethora of antibodies that recognize dystrophin and recognize the proteins that are associated with dystrophin. And he injected the purified dystrophin-glycoprotein complex into as many animals that he could get his hands onto. He injected it into hamsters, into sheep, rabbits, even a goat. And in fact, one goat in particular, his name was Goat 20, produced a wonderful panel of antibodies to all components in the dystrophin-glycoprotein complex, except the smallest component called sarcospan, which turned out to just not be very immunogenic. And he also took the gels of the purified dystrophin-glycoprotein complex, and he cut each of those protein bands out, homogenized them, and injected those into various animals to generate antibodies that he knew would recognize the protein that migrates at 50 kilodaltons or the protein that migrates at 42 kilodaltons, and so forth and so on. So that was really a great strategic experimental step that he did, which created a panel of antibodies that have been enormously valuable for the field of Duchenne muscular dystrophy. So he created not just polyclonal antibodies, which is what I was describing before, but he also created, in collaboration with Louise Anderson, he created a panel of monoclonal antibodies. Many of these monoclonal antibodies, as well as the polyclonal antibodies, are in use today and are also in use in the clinical setting. So these are really valuable reagents to have. Monoclonal antibodies have the advantage, even though they're a bit more expensive to create, they have the advantage of recognizing a single epitope on the polypeptide. So here is one of the validations that he published of two of the monoclonal antibodies that he generated, one to the protein that at the time migrated at 50 kilodaltons, and another monoclonal antibody that was created to the 156 kilodalton protein. And he is analyzing fractions from one of the purification steps to validate that these antibodies specifically recognize a protein that's associated with dystrophin and that is migrating at the correct molecular mass. He further validated these antibodies by analyzing skeletal muscle microsomes. So basically you can think of that as the starting material, the crude surface membranes, which was loaded in lane 1. Then he looked at purified dystrophin-glycoprotein complex, which is loaded in the last lane. And then he also looked at purified sarcolemma membrane proteins. Remember that dystrophin, we know, is located at the sarcolemma membrane. So this was a further validation of the purification step and a further validation of the molecular tools that he created. And again, all the proteins are revealed on the Kamasi blue stained gel, and the other experiments that are shown here are immunoblots. And in each of these immunoblots, the lab is characterizing individual antibodies to the 156 kilodalton protein, the 59 kilodalton protein, the 50, the 43, and the 35 kilodalton protein. And you can see that these proteins are enriched during the purification. And that's one of the hallmarks of protein purification, is that a protein is expressed in low abundance in the starting material, but its relative abundance is significantly increased during the purification. And this is a beautiful, beautiful example of that. And it really shows that, that all the proteins that they suspect are associated with dystrophin, because they know that dystrophin is enriched during this process of going from crude surface membranes to sarcolemma-specific membranes to purified dystrophin-glycoprotein complex. Dystrophin is enriched, and all of these other proteins are similarly enriched. That provides further evidence that these proteins are associated in a complex with dystrophin. So these antibodies were also validated by looking using indirect immunofluorescence on transverse skeletal muscle cryosections. And you can see these are performed with rabbit skeletal muscle, and you can see that as expected, as was published previously, that dystrophin is located at the sarcolemma membrane, and that all the other proteins, the 156 kilodalton protein, the 59, 35, 43, and 50, all those proteins are also associated around the sarcolemma, where you would expect them to be based on the prior experiment, right? The previous immunoblot demonstrated to us that those proteins were present in sarcolemma membrane purifications, and they were present in the co-purification of dystrophin. So this is a wonderful, wonderful validation that the antibodies are recognizing the proteins that are binding to dystrophin, that are co-purifying with dystrophin, and that are present at the sarcolemma membrane. So we know that dystrophin was associated and retained on the SWGA column. We know that dystrophin itself is not a glycoprotein. We saw that dystrophin is associated with many proteins of many different molecular masses, and that many of those interact with SWGA, which is a lectin that interacts with carbohydrates. So that's good and fine. Dr. Vasti and Dr. Campbell wanted to carry that a step further and look more specifically at some of these proteins now that they had the antibodies in hand, and look to see if these proteins were glycoproteins that could be that were decorated with N-linked carbohydrates. So they took purified dystrophin-glycoprotein complex and they incubated it with an enzyme called N-glycosidase F. And this enzyme will clip off N-linked glycans. And I hope that you can see that particularly for the 50 kilodalton, the 43 kilodalton, and the 35 kilodalton, that when we compare the migration pattern of the untreated DGC to the treated DGC, that those proteins shift in molecular weight and appear to be smaller. And that's because the N-linked glycans are released by this treatment with an enzyme that cuts off those, that digests away those carbohydrate molecules. So this again was the first demonstration specifically that some of those glycoproteins were decorated with N-linked glycans. And we're going to hear a little bit more about that later on from Dr. Baum. In addition, Dr. Campbell knew that dystrophin itself wasn't predicted to have any transmembrane domains, right? The primary structure of dystrophin did not suggest the presence of transmembrane regions. So that's fine, but when you look at the transverse immunosections, you can see that dystrophin is localized and enriched around the sarcolemma. Okay, so it's localized at the membrane. So if dystrophin itself is not a transmembrane protein, then why is it localized at the sarcolemma membrane? So to kind of get at that question, Dr. Campbell and Dr. Urvasti took purified dystrophin glycoprotein complex and incubated it with a probe called TID. And this probe partitions to the membrane bilayer, and it can be cross-linked so that it labels specifically any transmembrane region. And it turns out that this TID can also be radio-labeled with iodine-125 and can be detected by autoradiography. So showing you here are two side-by-side experiments. One is the Kamasi stained gel of the purified dystrophin glycoprotein complex. Again, you can see dystrophin, and you can see all of the proteins that are associated with dystrophin. And in the accompanying autoradiogram, you can see that there are specific proteins that are being marked with this TID probe that partitions into the transmembrane bilayer region. So you can see in particular the protein that's very hydrophobic or has probably many transmembrane domains is the 25 kilodalton dystrophin-associated protein. Many of those other proteins that migrate between 35 and 50 kilodaltons are also being labeled with this TID probe, suggesting that they also have transmembrane domains. So Dr. Campbell's beginning to build a model whereby dystrophin itself is not a glycoprotein, but it associates with glycoproteins. Dystrophin itself is not a transmembrane protein, but it associates with transmembrane proteins. And from this, they built a wonderful schematic model of the dystrophin-glycoprotein complex that is very, very similar to the model that we use today, which really reveals how accurate it was at the time. So Dr. Campbell's group was not the only group that was interested in purifying dystrophin and its associated proteins. Dr. Ozawa in Japan was also diligently producing purified dystrophin-glycoprotein complex using a somewhat similar methodology. And there was some competition, friendly competition at the time, as these two scientists investigated the dystrophin-glycoprotein complex and the proteins that are associated with dystrophin. So we've just learned how the dystrophin-glycoprotein complex was isolated using basic protein biochemistry. What other scientific findings can you imagine followed from the discovery of the dystrophin-glycoprotein complex? So the answer is shown here. This really was a groundbreaking discovery, similar to the discovery of the dystrophin gene itself. From the purification of dystrophin, scientists eventually, over the next 10 years, were able to identify all the transmembrane glycoproteins, and they named them alpha-sarcoglycan, beta-sarcoglycan, gamma-delta-sarcoglycan, sarcospam. They identified the dystroglycans, the alpha subunit and the beta subunit, and together they were able to construct this model revealing the function of this complex to link the inside and the outside of the cell and to protect the membrane from contraction-induced injury. In addition, while these proteins were being identified, their genes were also being studied, and it was found that mutations in any one of the sarcoglycans can cause autosomal recessive limb-girdle muscular dystrophy. Not only that, and you're going to learn much more about this later, but mutations that perturb the glycosylation of alpha-dystroglycan cause a host of diseases and myopathies, including limb-girdle muscular dystrophy, muscle eye brain disease, and congenital muscular dystrophy. And you're going to hear more about that later in the course. So this really was a discovery that led to many, many, many more other discoveries, which is really quite exciting. Okay, so you know now that dystrophin is not a glycoprotein itself. You can see in this staining of human sections that dystrophin and beta-dystroglycan are localized around the sarcolemma. And we know now that in patients that have Duchenne muscular dystrophy, mutations in dystrophin cause loss of dystrophin protein, which is shown to you here by the absence of staining in skeletal muscle sections from DMD patients, and that beta-dystroglycan, which co-purifies with dystrophin in the steps that we just went through in this particular module, is also missing from the sarcolemma membrane when dystrophin is missing. And that's another characteristic of proteins that are bound tightly in a complex together. Often when one of those components is missing, the entire complex is missing. And that indeed is the case, and it's very easy to see here for beta-dystroglycan. Caveolin 3 is not a member of the dystrophin-glycoprotein complex in the sense that it doesn't co-purify with dystrophin. And in fact, caveolin is not lost in the situation of dystrophin deficiency as we see in the DMD sample. So this really represents a nice example of proteins that are specifically lost because they're interacting with dystrophin in a complex and proteins that are abundant at the sarcolemma but that are not perturbed. Their localization at the membrane is not perturbed by the absence of dystrophin. So that concludes our module on the discovery of the dystrophin-glycoprotein complex. I hope you enjoyed hearing about all these fun and exciting biochemical techniques to purify protein complexes from skeletal muscle. And I hope in particular you have an appreciation for some of those initial experiments, how critically important they were for really changing the direction of the field. And what I really like about it is how accurate those experiments have been, and they've really stood the test of time. So I hope you've enjoyed it.